is that you can't find genetic associations in smaller samples. You could actually look at all kinds of disease-related questions. So you could say, well, I've heard that schizophrenia is associated with brain abnormalities. And I read the newspaper and I can't make sense of it. It's just all sorts of different things, different structures all the time. And I've heard that bipolar disorder has a milder phenotype on the brain, with depression maybe even milder. So some of the studies that we talked about that Enigma's doing that measure MRIs in a consistent way can ask similar questions. What are the patient versus control differences for various things that we measure? And this can provide some answers to questions that have been a bit of a mystery for a long time. So we were publishing about 15 years ago brain deficit schizophrenia. You could run it again in a different cohort and not find the same brain deficits at all. So these are very, very large studies, large numbers of patients, large numbers of controls. They're intended to find brain measures that are the most different between patients and controls. And they then become targets for genetic analysis. There's no point measuring. So for example, the intracranial volume, the size of the head, isn't different in schizophrenia versus controls. But certainly the ventricles are bigger. These structures are a little smaller, but not much, which explains why it hasn't always been found. And because these patients take medication that is trophic to the basal ganglia, these structures are all larger. So this has really taken off, and you can look at the agreement across sites around the world in brain measures. Ventricles are bigger in schizophrenia in these 4 cohorts. They're not bigger in this one, so you look for errors. It's actually kind of useful. So I'll just move on a little bit. Doing this kind of thing in a systematic way obviously would allow you to do diagnostic classification, or cyst diagnostic classification, for disorders that have traditionally had symptom profile overlaps. All right, so last bit of the talk. Are we doing okay with time? Oh, we're doing okay. All right, talk will be over in 5 minutes. So if you go to a brain mapping meeting today, you'll see that the thing that gets people the most excited probably is the ability to map brain connectivity. And diffusion tensor imaging, this is an image by David Shattuck at UCLA, he was at USC. And he's looking at 7 Tesla diffusion MRI. And this is the kind of scanner that will be available at USC, we hope by the end of the summer. And these types of images would be available for people that want to study them. So you could say, well, how do I know if this person is ill? I mean, what are the metrics? Do we measure the geometry of the tracts, or the integrity, or something about the networks? Do we put them into pattern recognition algorithms like the other things? I mean, you can see the phenomenal detail. So this is rendered by software that Richard Leahy and David Shattuck have developed, which is incredible. So this is the richness of data shown in a software package called BrainSuite that Richard and David and others have been working on. And it presents you with an interesting dilemma. So again, this isn't the standard DTI scan. It's one from something called 7 Tesla hybrid. It's hybrid diffusion imaging. I'll tell you a little bit about what that is. So in an MRI scanner, you can send in a radiofrequency pulse that will excite the quantum state of the protons in your water molecules into a higher energy level. And the rate at which they relax back down to the standard energy state is determined by the type of tissue that they're in. So that's how an MRI works. It was noticed about 30 years ago that if water was diffusing through the tissue, the MRI signal was relaxed or decayed much quicker. And partly because these magnetized spins were floating out of the box the way you magnetize them. So the principle of diffusion imaging is that you can do an MRI scan, and in a voxel, you can set up the MRI protocol to tell you which way the water is diffusing. And by connecting these up across a whole scan, and there's algorithms to do this, you can make an inference about what parts of the brain are connected to which other parts of the brain. Now, you don't know if it's correct. You don't have access to the post-mortem data. But at least in the cases where this has been validated, you can scan acrylic phantoms that have plastic fibers. It seems to get it right the majority of the time. So how do you measure brain connectivity? Well, the standard way that brain imaging people do this with structure, functional imaging is a little bit different, is they'll track all the fibers that connect 2 regions of the brain that we know are connected. So I mean, we know the frontal lobe and the parietal cortex are connected. This might be a topic of interest in studies of inhibition or studies of adolescent development when the frontal lobe isn't completely done developing. And you can ask questions. You can say, well, what is the density of the fibers here, or their integrity? Now, the nice thing about looking at this is you don't have to focus on one tract at all. So let's say there are 70 regions in the cortex. I don't know how many regions there are. Let's say there are 70. This is a oscillation. You can say, I mean, just like different cities in the US, whether there are freeways between them, you can say, well, tell me if there are any fibers, or what is the density of fibers between this region and this region. Now, if all the regions in the left hemisphere are along here and the right along here, and the same ones are listed again, if anything's connected, you can put a little count on that. So you can say, well, I've noticed this fiber goes through the brain. Can you add a count between this region and this region? You can compile a matrix. And so you can think of representing brain connectivity using a 2D matrix. And it's basically like a road map. And these are cities, and these are whether there's a freeway that runs between them. So how would you do a genome-wide scan of the brain's connection? So we know that the way the brain is wired is partly due to a genetic program that plays out over time. It's certainly not impervious to the environment, but there's a certain genetic program that guides axons to the regions that they're supposed to be in. Now, this is work again by Neda Jahanshad, who's here, published in PNAS earlier this year. If you screen the genome on every connection in the brain, you have this sort of ridiculous statistical correction again. And so let's say there are, I don't know, 70 regions by 70 regions. That's 49, let's say it's 5,000 connections. 5,000 times a million SNPs in the genome times your 20 to 1 correction for alpha. You're going to have to find a genetic association somewhere in the brain that beats a 10 to the minus 9 threshold. And with the samples we have today, that's very difficult to do. So one of the things that came from the power study earlier is this notion of looking at power for searching for gene effects in images, is that if you look at heritable connections, in other words, ones where there's evidence that siblings are more alike, or twins are more alike, than randomly selected people the same age, then you have a better chance of finding genes that affect them. So this was a screen of the connectivity that would discover whether a connection is heritable. In other words, if you have a brother or a sister who's your age, is the integrity of that connection, or is the density of that connection more similar to you than if you just picked a random person who had the same age and gender? And it's variable. So some of these connections are either not heritable, or they're so difficult to measure that you couldn't detect heritability even if it's there. So this brings up an interesting idea. So you could do a connectome-wide, genome-wide screen. You could say, well, I want to go through each of these connections, and maybe there's that's it's supposed to be the language pathway, it's Broca's area, it's the Wernicke's area. And you could screen the genome for things that look like they explain some of the variation in the database in these different areas. And in Neda's work, she found a gene that, in fact, about 3 weeks later was discovered to be an Alzheimer's risk gene. So that's very exciting. So the psychiatrists, some of the skeptics would say, images are never going to be useful because they're such an indirect evaluation of somebody's symptoms. It's much better to look at the genetic associations with people's symptoms because one causes the others or whatever. So there's a little bit of a bet on the table as to whether images are going to be more efficient in the long run for finding how genes affect us than just by looking at behavioral differences. But a nice synergy that happened with this paper is that the gene that was implicated called SPON1. 3 weeks later, Robert Green from Harvard published a study in which that was the top gene that was associated with measures of cognitive function in people who are elderly. So I mean, that's just too improbable to be a coincidence that the same gene would be picked. So just sort of to finish this up, I'm going to end with a mathematical problem. So many of you are very, very accomplished mathematicians. So neuroscientists always want to find measures that are informative. You want to find something about the brain that is going to tell you about the behavior or disease that you care about, or is going to be linked with some genetic area that is of interest. Now, we said a moment ago that you can split the brain up into components, and there are some agreed ways to do it. Hannah and Antonio have developed atlases. So atlases of the brain split the brain into regions that anatomists regard as functionally separate, or separate for cellular regions. So what we do when we look at connectivity is we sort of break up the brain into a certain number of regions, look at connectivity. This induces a network. So I need to explain a little bit how this is isomorphic, has the same information as this. So two different parts of the brain, this and this, can be represented as two different nodes in a network, this and this. And if there is a connection, you can connect them. Now, not everything's connected to everything else, and even if it was, find the connections. And so there is a sort of basic network organization in the human brain. And from this network organization, you can compute metrics. You can look at its efficiency. You can look at something called the rich club. So the rich club is a tendency for certain nodes in the brain to be much more highly connected than they would be by chance alone. There's a phenomenon called the K-core. K-core in a network is something where if you threshold the nodes by the number of connections, you only keep those that have more than K connections. This is supposed to be a much more efficient and clearer picture of what's happening in the brain and looking at all these connections. So this has become very popular. And if you make these networks, you can compute small worldness, clustering, efficiency. There's even toolboxes that allow you to do this. So let's say you can compute everything you can think of that might be informative about the brain, topology, wiring, whether there's a connection. Plug it into one of these classifiers. So we talked about Lasso, which is a machine learning method. Another one is a support vector machine, where you have data and you're trying to classify two groups. So you can say, well, I'm interested in schizophrenia, but I don't really know what it is about the brain that's different. I have some hypotheses, but can we have the machine try and figure it out? So one of the ways that you can do this is written by a postdoc, Gautam Prasad, and it's a really nice method. It's clever. So let's say we pretend we know no neuroscience, which is not quite right. You can partition the brain into a large number of randomly segmented regions. That doesn't look like there's any coherence. You could examine the connections between those brain regions with tractography like we looked at before. And you can plug the connectivity matrix into a classifier to distinguish patients from controls. Now, this could never end. I mean, you could do it again and you get another answer. But there'll be some parcellations of the brain that are better able to distinguish things that we care about than others. So a parcellation that just has two regions and cuts the brain in half might be able to pick interhemispheric communication disorders up very well. But it sort of affects, a little island of the speech area in the left hemisphere will have a different answer. So this is a very heavy computation. And it's related to, my daughter said, don't show this slide. And, it's related to something that mathematicians were thinking about at the turn of the last century. So very brilliant mathematician, Srinivasa Ramanujan, who was working in England. And he was fascinated in the early 1900s about the partition problem. So the partition problem is, if you have a large number of things, how many different ways can you sort them into groups? Well, there's a ridiculous number of ways. So this is something called, these are Genji symbols, if you've heard of those. So there's a Japanese novel called The Tale of Genji. And each of the chapters of the novel has one of these symbols. So if you're in chapter 12, you have this little symbol there. And what it means is, if you have 5 things, this little pentagon here, you can group these two together and these two together and leave that one on its own. Or you can group these 3 together and leave those 3 on its own. And this is a way of organizing the ways you can group objects. It's a very deep mathematical idea. And one of the things that Ramanujan was interested in, and later a mathematician called Bell, this is called Bell's number, is how many ways there were of dividing regions into groups. And it's just this fantastically large number. So for 5 things, there are 51 ways. So the 5th Bell number is 51. I'm sorry, 52. So you get a combinatorial explosion when n is large. So let's say you were trying to group brain regions into consistent areas or informative regions. You'd have to search for a long time. Even with all the computers in the world and our entire lifetime, you'd never be able to search all of the 10 to the 189 combinations. And this is the recurrence relation that defines the nth Bell number. It's very largely quicker. So what Gautam did, and I'll send you his paper, it's pretty good, it's not out yet, it's here, is to think of a way that you could search the set of all the partitions of the brain in an efficient way where you could find ones that are better than the ones that we have today. So one example is FreeSearch. Many of you guys have used this software. It's very good. It's written at MGH. It splits the brain into islands of the cortex where we look at the connectivity between one region of the cortex and the other. But if you run EPIC, this evolving partitions in connectomics, you are better able to distinguish or classify people, in this case with Alzheimer's disease versus controls, than if you use the standard partition. So random patches are not very good. EPIC's pretty good, not perfect. It'll still get 19% of people wrong. If you have a group of Alzheimer's patients and healthy elderly people, and you look at their structural connections, get 81% of people correct in terms of whether they have Alzheimer's or are healthy, and 91% of people wrong. The wonderful thing about that is that number goes up every day. So the longer this runs, the more connections you find that are better able to differentiate illness. And it's just a wonderful problem. You just keep updating the paper with a better number in there. So that's a sort of mathematically intractable problem that we can watch being solved. So just to wrap up, what is the point of doing ENIGMA? Well, it was originally a genetics project where you can screen brain images worldwide to find genes that affect the brain, and in some cases disease risk. So not all the genes that affect the brain have anything to do with disease, but you can check when you look at databases. Partly through the work of Nana Jahanshad in my group and others that are looking at DTI, we are discovering genetic variants that affect brain wiring, and some of them are also disease genes. And so there's this bet on the table with the genetics people that imaging will somehow be more efficient at finding interesting genes than screening diagnosis. It doesn't look that way. It looks like you probably need as many images as you need people in a diagnostic study to find genes that affect the things you're interested in. It's also feasible to do genome-wide search throughout an image or throughout a connectome. Some people have called this big data squared. So there was an NIH call for applications on big data. I think that word is getting used rather a lot. To get into those problems, you need a lot of clever mathematics. And so a lot of the work that you see in sparse machine learning. So machine learning will try and find patterns between predictors and outcomes. Sparse methods will try and penalize solutions or favor solutions that are more efficient. So this notion of sparsity, you'll see this coming up quite a bit, as the amount of data available gets much bigger. You can do data reduction in the images or in the genome or in the connectome. So you saw a study where the heritability of the connections was used as a filter. You threw out connections that weren't heritable. So we're only just at the beginning. So this is not the beginning of the end or the end of the beginning. It's the beginning of the beginning. We're experimenting with distributed computation, partly because some data is private. Now this is very unusual. If you go to NIH and you say, well, I don't want to share my data with anybody. The NIH won't be very pleased because there's a mandate that you have to share images and genomes that you collect. But there is a privacy problem in sharing some genomic data. If you're able to identify the individual it came from and the illnesses that they may be likely to have, that's regarded as a more serious problem than not sharing the data. So some of the solutions and ideas are very welcome, require computations on remote data sets that we're never able to aggregate all of them in one place. So thanks very much to the folks in the IGC, the USC Imaging Genetics Center. Derek's here today, thanks very much. Nada's here today, she's at the back there, thanks very much. Lots of data aggregated from Australia and Queensland, folks in the Netherlands. And really, the only dilemma with these is that assembling the author list takes a little longer than writing the paper. So trying to get these papers finished can be a bit of a challenge. So I appreciate your time. Come and see us. We're at the marina and we need